Good morning, good afternoon, good evening, everybody. My name is Luk Vandenberghe. I'm the Grousbeck Family Chair in Gene Therapy at Mass Pioneer here at Mass General Brigham. I will be discussing with you our work on decoding and recoding AAV towards a future of what we refer to here as rational AAV design. Just maybe to set the stage a little bit, we make a statement here is that the potential of AAV has been proven, however, is meeting its technological glass ceiling. Most of you are well aware that this modality of gene therapy is really an emerging one in 21st century medicine. AAV, at least for in vivo gene transfer approaches that are intended to be durable, is seen as a vector of choice technologies. However, with increasing number of clinical readouts, we are reaching the limitations of the current state of the art in that technology, both in the kind of safety parameters as well as in the efficacy parameters. There have been a number of very attractive vector engineering approaches that have been belabored over the past decades or so. However, the observation is that very few of the results of these engineering efforts have been adopted clinically. And we asked the question here, why that is, and we try to, in that gap analysis, try to address some of these shortcomings. So these shortcomings ultimately have led to a proposition here that we aim to rationally design AAVs. And now the question is, is there a need to further the technology in this space? Well, the number of questions, some of which are listed here, that have been asked to the field for the past 20 years, including myself, have been questions like this, right? How do we detarget, for example, from the liver, which we know is a primary sink, when our therapeutic target tissue really is the CNS or the muscle, such as in Duchenne muscular dystrophy? How do we administer intravitreally when targeting the retina? Much more clinically amenable than a subretinal approach, which is currently the approach used in Luxturna. And one goes on, ultimately to reach even questions that take into account some of these novel genome editing modalities or more complex modalities, such as tissue specificity that is introduced by complex promoters or regulated promoters. Here, how do we package Cas9, a guide RNA, and an HDR cassette in one vector, and ideally then give it hit and run pharmacokinetics? None of these questions we've been able to address over the past decades, and we've been able to improve upon with the engineering efforts that I spoke about earlier. These are real questions. They show up on, for example, here, the label of one of our first approved drugs, Zolgensma, where liver toxicity is clearly seen as a potential serious side effect of that otherwise transformative drug. So how do we govern this? Well, how do we think about this? Well, most of us are looking at this turquoise halo on this buckyball particle that we refer to as adeno-associated viral vector, or AAVs. That halo here really defines efficacy, safety, and manufacturability within this space. And there's been a significant precedent set that indeed slight variation on this capsid structure does have significant impact in all of these categories. For that reason, many people have explored diversifying the capsid portfolio that we use. And initially that was by natural serotypes. Some of that effort I was part of about 10, 20 years ago that really expanded massively. And indeed, most of the vectors that are currently moving to INDs or are in INDs or in products are ultimately derived from these natural serotypes. The engineering space really is minimal in terms of adoption into INDs. That engineering space is really broken up in kind of two main categories. The one above here, the directed evolution one, unbiased approaches at creating large diversity that then is screened through, often in an iterative selection scheme. That has been the main foray. However, rational design also has been done at a much more limited level. But I would argue neither one of which has been successfully adopted clinically. What we have developed is really, again, this gap analysis where we, in a kind of layered and tiered approach, try to identify the limitations of the current methodologies. And what we came up with is, instead of unbiased libraries, we designed curated libraries. We call them smart libraries here. They're really curated to maximize functional diversity. And I'll give you one example of that in one of the following slides. That helps us narrow down the search space and gives us much more likelihood of actually finding that needle in a haystack. To help us even more, we attached a barcode to it, illustrated here. That barcode now enables a screening modality that's exhaustive, quantitative, and multiparametric. What that means is really with next-gen sequencing, we can get incredibly rich data sets out of this in a methodology we call AAV-seq. And the data that comes out of this really informs the structure-function relationship of AAV much more. AAV generally is still seen as somewhat of a black box therapeutic, and we hope here to shine a light on that structure-function relationship. That information can then be leveraged to make libraries iteratively better and more defined, but it can also further us in terms of getting to rational design elements that allow us to tweak these vectors in very specific ways. Here's an example. We used evolutionary information to get to what we call ANC library, ancestral AAV libraries. These are these curated libraries I was talking about earlier. We barcode them, we quantify the input of a particular screen, and then we take screens into in vitro models, mice or non-human primates. And here we look at things in a multi-parametric way. Even though we are maybe primarily interested in, say, the liver, we still have full biodistribution as well as bioactivity data in all of these other tissues or all of these other models. What does the data look like? Very briefly here, we can candidate select out of this library. What is illustrated here is one plot of a library with 2048 variants. Each dot here represents the properties in one of those parameters, in this case, the liver, of each of the members of this libraries. And here you can obviously select your best liver vector or your worst liver vector, if that is your interest area. In the meanwhile, that same data can be converted to what we call a fingerprint plot, and that shows us really the defining features that illustrate why there are some vectors that are more functional in the liver than other vectors. And here we can see that there's a motif, this position tree effect, that drives this particular liver biology. We can take that information and we can now take a vector such as AAV9, deprogram it from the liver, not just in mice, but we can do that in non-human primates. And here we can take, on the opposite side, in a gain of function study, a vector like AAV3B, currently already in the clinic for hemophilia or a similar vector that we can then further improve upon by converting this liver biology that we have observed in our screens. So what is the differentiated opportunity here? Well, the goal is really to ultimately unlock either novel therapeutic areas. And in fact, we've done that once through this asset already. We've worked in the cochlea for hearing and balance disorders. And there we found that within the libraries that we've defined of these synthetic curated variant libraries, we've identified one variant that particularly targets a cell that previously was inaccessible. And that has opened up a space in and of itself through one of the spin-out companies of Mass General Brigham called Akouos, now a publicly traded company. Second, what we hope to do is within each of these categories, some of which are already emerging spaces, such as the CNS, we hope to obviously expand the therapeutic reach of this modality. In summary, we have a very large functional and barcoded library tool here that is continuously expanding, that allows us to rationally design AAVs. These are a defined track record in terms of IP that is quite strong. It allows us to screen multiparametrically, really trying to define the in vivo pharmacology of these agents. And we have a number of vectors at hand which we feel are lead candidates. The opportunity and outlook for our work is really to do more cost-effective and informative AAV screening, work towards that rational design that cuts out eventually the screening. We've commercialized and at some level are looking at doing first-in-human studies through some of our licensees, and we seek further partnerships in gene therapy. Our current work really is emphasizing improvements on AAV manufacturing. So with that, I look forward to the discussion and your questions. Thanks so much for your attention.